Um, Peter Barham is our second speaker this evening, and Peter is speaking to us from Cape Town this evening, where he's an honorary research associate in biological sciences at the University of Cape Town. He is from Bristol University in the UK, and I've asked him just to tell us a little bit about how a physicist can turn his attention to African penguins, and so I think he'll have some interesting stories to tell us. But Peter has a great love for penguins. Not only that, he's also authored the book The Science of Cooking. So gee, Peter, you sound like a very, very interesting person indeed. Thank you so much for joining us this evening to tell us about how the ways of marking African penguins have changed through the decades. Peter's talk this evening is titled, "Which Penguin is Which? From Steel Bands via Rubber Bands and Spot Patterns to Transponders." Peter, over to you. Thanks once again.
So, penguins and how you mark them. Well, how did I get involved? Let me begin by telling you a little bit about myself. I am, as Nick said, a physicist, and that's what I really do for a living. Well, I don't do anything for a living any longer because I'm retired, so I can do whatever I like, whenever I like. But I first got involved with penguins when I met my wife. That's more than 50 years ago now, and she was in love with penguins from a very early age. They're her favourite animal, so it was very simple to keep up with her because all I had to do was buy her a book on penguins, take her to the zoo to see penguins, buy her a penguin t-shirt, whatever, and that was all I had to do to be keeping her good books for some time. But after some years, there's nothing else you can do really. And if you ask Les, who knows me reasonably well, he will let you know that everything in our house around us has penguins in it somewhere. So eventually we decided that we would have to go and see penguins in the wild. We started doing that back in the mid-90s when we realized it was actually possible to do that sort of thing. And then whilst doing that, we came across an advertisement for the 3rd International Penguin Conference, which was being held in Cape Town in 1996. And we decided that would make a really nice holiday. So we turned up, having first checked with the organisers they would accept rank amateurs who knew nothing about penguin science to attend their scientific conference. And during that conference, there was, well, the first thing I found about this conference was it was a biology conference. It's the first biology conference I'd ever been to in my life. I've been to many, many physics conferences, and the contrast was enormous. In a physics meeting, if a student were to make a rather silly presentation or to make some stupid remarks, they would have been ripped to shreds by the senior professors around. It's a very robust business, physics, and we can be, or certainly in those days were, quite aggressive. Biologists, on the other hand, were not at all aggressive. There were some talks that even I, as a rank amateur, could see there were some really big holes in them, but nobody said anything. They were all very polite. Until a chap called Bernard Stonehouse, who was one of the senior penguin biologists, the first person to actually really spend a full winter studying emperor penguins in Antarctica, decided to change the subject of his talk completely from that which was advertised and started talking about the way in which penguins were banded with steel or metal flipper bands, and that process had been discussed at the previous International Penguin Conference 4 years previously, and there had been a consensus then that it was not a good thing to do, it was harming the penguins in some way, I'll talk about that later, and that people really should get on and find a better way. And he, toward the end of his talk, suggested one such solution might be to use plastics rather than metals to mark the birds. Now, it turns out that my profession, my research was on polymer physics, which polymers are a fancy name for plastics. So my wife kept digging me in the ribs to say something, as all these biologists, penguin people, started really ranting at each other about why plastics were perfect or plastics couldn't possibly be made to work or anything. So I decided I would have to say something. So I owned up that I did in fact know a little bit about plastics, and that there could be possible ways of improving using plastics that would have to be looked at. As a result of that, the best way to put it is probably volunteered to go away and do something. And I will tell you a little bit about what we did later on. So that's the background how I got involved with penguins. Then it became much more than marking penguins. So, but anyway, let's talk about penguin marking first of all. And the first question I should ask is, well, why do you mark penguins? Who'd want to? And the very first marking of penguins that I can find a record to was in 1908, when Louis Gahn, who was a French biologist, used some coloured celluloid tarsal or leg bands, which were meant for chickens, on juvenile Adelie penguins, and the idea was to see whether they would return to their natal colony. This was a very surprising piece of research, actually, because up until that sort of time, biologists in general did research, particularly on penguins, simply by collecting specimens, usually by shooting them and then cutting them open. That was the normal. This guy decided he wanted to find something out, quite specifically, where do they go and when do they come back? So he put these rings on them. Now, in practical terms, celluloid is not a good material for doing this sort of purpose. It's not very robust. It certainly doesn't like any much in the way of temperature cycling. So he didn't get very many returns. He didn't really get much results out of it. The next person who really did some sensible marking was a chap called Murray Levick. Murray Levick was a New Zealand/British naval officer who spent some time at Cape Adare, and he studied the Adelie penguins there, and he made some superb notes on their behaviour. Probably the most important part of his work was actually completely suppressed for about 100 years, because what he was looking at was their sexual behaviour. And what he found, was so outrageous, the way in which these penguins behaved, that it could not be published. And in fact, it's only in the last 10, 15 years that the pamphlets have been rediscovered, and the information he found about homosexual behaviour in penguins, necrophilia in penguins, rape in penguins, extramarital sex, all those sorts of things have actually come to light, and they've all been established properly by modern scientists. And it turned out that he'd done all this work by about 100, maybe 80 to 100 years before it was finally published. He did his work by painting the penguins red, not with any banding at all. Then the first real study of what you might think as modern penguin science was by Rance Richdale, who lived in New Zealand on the Otago Peninsula. He was a school teacher of geography, but he had an interest as an amateur ornithologist, and he decided he would follow the behaviour breeding cycle of the yellow-eyed penguin colony, which was within walking distance of his home. And he was the first person to use what you might call proper rings. He made tarsal leg bands from aluminium, and this is a photograph of his actual kit that he actually used, published from one of his books. And he had both aluminium rings that were numbered and celluloid rings which were coloured, the celluloid rings being used on chicks to identify them. And these are all on legs. Now, there's a problem with that. Here are a couple of pictures of yellow-eyed penguins that I took some years ago. And what you can't see is their legs. So if he wanted to know which bird was in a nest, he actually had to go to the nest, put his hand in, pull the penguin out and look at its leg, and then put it back again. Now, that's probably okay with yellow-eyed penguins. If anyone tries that with African penguins, you know that will be an incredibly bad idea. But it stresses the birds anyway, so it's not a good idea. And if you look at some penguins in general, here are a few penguin pictures. And how many of those birds could you actually see a leg band on? The answer is just this chin strap over here, and that's because he happens to be standing so that you can see it. If he was at his nest, you wouldn't be able to see his leg at all. So leg bands are not really suitable for penguins. And it wasn't long before it was realised that that happens, and flipper bands are better. Flipper bands are quite nice and easy to read if they're put on, because you can see here's an African penguin with a flipper band, just there. This particular penguin, I read his band with a telescope from a distance of probably about 100 meters. And then we got a long lens out and photographed it so you can actually see a photograph to prove it. For those of you who were around at the time, that is Peter the penguin, the famous treasure bird. And that's the only time he was seen after he was released and found back again. But they are useful things. There are also problems with flipper bands. While they're easy to read, they're also easy to misread. In a survey we did some years ago with volunteers, when we were comparing different volunteers reading the same band, we estimated that the accuracy of flipper band reading with a telescope was somewhere between 85 and 90%. So you get a lot of misreads, unfortunately, as well. Not as good as they might be. But they also do other things to penguins. They increase hydrodynamic drag. If you're wearing a rattly little bit of metal around your arm, you try to swim, you'll need more energy to swim than if you didn't have it there. That was discovered, well, measured properly, I should say. Discovered, it's obvious, but it was measured properly by some German and British scientists working in Antarctica, where they actually caught Adelie penguins and put them into tunnels which had to swim through. And then they measured the oxygen use while they're in the tunnels to show that if they were wearing a band, they used more oxygen if they weren't wearing a band, and calculated that there was something like a 5% energy penalty to wearing a flipper band, which matters because you've got to catch so much more food. But more importantly, in my opinion, is the fact that the metal flipper bands actually damage the penguin's feathers. These are some photographs of African penguins. They're all four different African penguins. If you're wearing a metal flipper band, you can see here there's a fairly deep cut underneath the wing. Here there's sort of a line of feathers have been cut away. Here there's a bit that's beginning to wear away. And here is a fairly bad example, but not uncommon, where there's a whole patch of bare skin. And if you remember, penguins, particularly Antarctic penguins, swim in rather cold water. And part of the purpose of their feathers is to make a waterproof layer that prevents the cold water reaching their skin. So when a penguin has this sort of amount of bare skin, then it will need, particularly in colder climates, to use a lot more energy to be able to keep itself warm, and that is a serious penalty. So that means, that's probably one of the reasons why it was found, several different studies, that penguins, particularly in Antarctica and the subantarctic, have a poorer breeding success and lower survival rates if they are fitted with bands. But often the better studies were where they had better, I should say, is possibly not the fair word. The studies which are more reliable are ones which they fitted not one but two bands to some penguins, and the double-banded penguins were far worse off than the single-banded penguins. Now, why does this damage occur? Well, first of all, the metal is harder than the feathers, so there's just a rubbing of metal against feather, it will wear. But more importantly, there are sharp edges on the bands. This is a flipper band, this is an African penguin flipper band. You can see there are corners. This is a set of 3 flipper bands, all of which were closed properly, like that one, and then cycled in temperature. So all we did was we put them into iced water for a couple of hours and then put them into an oven at about 30 degrees for a couple of hours and repeated that 100 times. And what happens is that, as with all metals, and as every material scientist could tell you, that's why I knew about it before I started doing the experiments, that metals are not static things, they are dynamic. They will change shape if they are cycled in temperature and so on, unless they were properly annealed when they were formed. So the way to make a flipper band which would not change its shape after it's been bent to fit the penguin would be to take a blowtorch to it and hold it at about 120 degrees for about 10 minutes. That might kill the penguin, but of course the band will then stay closed. But if you don't do that, then what will happen is the band will open and close a bit. So you can't make them without sharp edges, so they're going to end up causing damage. The other thing, which is less frequent but really unpleasant, is that penguins wearing flipper bands can get trapped by them. The band has to be loose enough that the bird can molt without cutting the blood circulation when it molts, because on molting the flippers swell up a great deal. So that's why the shape of them is what it is. But that means there's a hole, a gap between the flipper and the band, and a penguin really doesn't have a reverse gear. So it walks through some undergrowth, and a twig or a branch can slip between the ring and its flipper, and then it keeps on walking and it gets trapped. So if you look closely, just here, this bird is wearing a flipper band, and it is caught it on this branch completely. So the bird is unable to free itself. That I've found myself on Robben Island quite a number of birds which are dead, just left hanging by a flipper band. But this particular bird was lucky. We found it probably no more than a day or so after it had been trapped. We were able to free it and then send it off for some free fish at Sankop up the road, so that it was then recovered its strength and was sent back to the wild with the band made a little bit better shape, but still wearing the band. So although we can get fantastic information from these flipper bands, we know, first of all, we know that they are damaging the birds to some extent, some birds more than others, which also tells us that it's quite likely that the data we acquire from them, things such as lifespan, survival rates, if you like, nest faithfulness to their partners, anything like that, is liable to be not quite as good as it should be, because the birds have been affected by the very fact they've been marked. So what we did after this to try and help was to design bands which would be less harmful. And conclusion first we did was that a soft rubber, or a softer material, and rubber's probably the best material, but we tried a variety of different materials, is going to be less harmful. So we thought, okay, fine, we'll design a band. And we began by doing really fancy, crafty things. So we designed a band which would actually, as it swam along, as the bird swam, got faster and faster swimming, the flipper was going to come out through this gap here. And this shape is designed so that if the penguin is swimming along, the band will move up the flipper and hug the body. So it can just becomes part of the body, and it's soft enough to contour the shape of the body. In tests in a model penguin in a tank, there was a tiny increase in drag compared to other designs. I mean, it was something like 0.5% increase in drag compared to like 10 or 15% increase in drag with some other metal bands. And that's fine. But actually, in reality, it doesn't work very well. So we then decided to be a little less ambitious, and we did things like having a spring-loaded band to make sure that it didn't spring with a flexible rubber spring inside, just to make sure that the band didn't have any rattling on the penguin. Again, it works fine, but it didn't really have a great benefit in the wild. Tested in the zoo, they're great, but in the wild, not so great. So in the end, we just came to plain laser-engraved silicon rubber bands. So that there's a ring of rubber, which is the same, similar shape to the metal ones, but a bit smaller, which just simply stretches over the flipper. So the putting the band on is a much less stressful process for the handler and also for the penguin. And putting a metal band on a penguin is a stressful process, because penguins don't like being manhandled. They don't like people doing things like that to them. And African penguins are notorious for being, shall we say, vicious. If you pick an African penguin up, you can guarantee you'll get bitten, scratched, everything else. So you have to be very firm with them. And one of the good descriptions I had from one of Les's ex-PhD students was a penguin is a pair of razor blades on legs. And that's basically what they feel like. So being able to put them on easily is good. So these bands all worked very well on our zoo tests. We tested everything out, first of all, in Bristol Zoo, before we came down to South Africa and tried them on birds on Robben Island. And we finally got to one that was good for zoos, and in fact is now the standard zoo banding for penguins. But what we found was they fitted on the birds beautifully. They were easy to put on, great. But within about a month or so, the penguins also learned how to take them off. So what we ended up with was the bands wouldn't last very length of time, so actually they were useless. So I'm afraid we had to abandon completely trying to find a better way of making bands, because anything we made that was going to be helpful ended up the penguins decided to take them off. So I moved on to another idea, which was let's try marking penguins, let's identify penguins without marking them, without doing anything to them at all. You can do that. I realised this, and it was interesting to see that the penguin biologists in South Africa didn't realise the same thing, that the penguins have a unique pattern of spots. Every penguin has spots, every African penguin has spots on its chest, and those spots are different for each bird and don't change. So here's a group of penguins from Bristol Zoo, and if I overlay these symbols on them, you can see that this bird and that bird the same, that bird and that bird the same, and that bird and that bird the same, and others all different. And in fact, zookeepers regularly use, and have used for years, the individual spot patterns to recognize their birds, because you're with those birds day in, day out, so you very quickly get to recognize them. And you know, I knew that, and I knew that I could recognize the birds at Bristol Zoo. And just to prove it works, I took some photographs of some birds here in South Africa in different years when I was out visiting, and as you can see, there are birds with very distinctive patterns that you can easily pick up time and again. And here are some pictures from different years of some of those birds. So you know, the pattern does not change when the birds molt. Although now we've done a little bit more work on that, we can say that there are small differences. The thickness of the band changes slightly, so some spots might get trapped in the band. So there may be small differences, but they still remain pretty much unique. Well, they do remain unique as far as we know, but they remain pretty much constant. That's not quite so constant. So what do you do with this? Well, what you do is, if you're a physicist, you say to yourself, I want to be able to do this automatically. I don't want to have to spend hours peering at photographs. And to do that, you need to go and find a tame computer scientist. Fortunately, at Bristol, I knew a few tame computer scientists, so I went and talked to one, and he then found a PhD student who could start doing some useful things. And this is what we came up with. This is a system which we built, which worked on Robben Island for some time. Basically, it's a camera, or camera system, which connects to a computer, which then takes images on a regular basis, and we can use it both at day and night because we use both infrared and visible light. And in fact, we even have on here a infrared light source so that we can illuminate it very much better with uniform illumination sometimes. And this was left on Robben Island over a couple of years. This is Rich Shirley, who was a PhD student. He's not a computer scientist, he's a biologist, but he was working with our computer scientist, Thilo, and they built this thing. Okay, now I'm going to show you how it works after a fashion, but just to give you an idea, these will be penguins passing the camera. And what the system does is, as soon as it finds something it thinks is a penguin, it draws a green rectangle. Well, it doesn't draw a green rectangle really, but as an illustration that I know that penguin's identified, we've superimposed a green rectangle on the video. And then once it's gone to its database and said, aha, is this a penguin that's already in my database? Can I be sure of it? If it's sure, it draws a yellow rectangle around it. So that's an identified penguin. So now I'm just going to quickly swap the screen share. So now you can see the sort of things that are happening. You can see some of these birds are banded and some are not, but they've all got spot patterns, and most of them get picked up as being penguins, and most of them get recognized eventually. So that's what the system is capable of doing. I will play that again so you can get a second look at it. And while I'm doing that, I will say this is not a real-time video. This is a video taken which was post-processed to achieve this. And that's important because it tells us something about limitations of the system. So basically, if you're going to do this, you're going to have a system which is going to work. You have to do, in some order, find the penguins in the images. That was this little green box. And that is the thing that was done in real time, pretty much. So basically, it finds a penguin. And what it does when it finds a penguin is it just cuts an image out around the penguin. And these are examples of images which were cut out around penguins using that system at different times. These ones are coming from the sea, these ones are going to the sea. The system, we could turn it around during the day. Then after that's happened, because you haven't got time to do it with computer time online, you have to do various things. So the first thing you have to do is normalize the lighting, so you actually get them all lit similarly. So they become grayscale images with just the penguin highlighted, and that's the next stage in the processing. Many images you can't do that to because there's too much contrast or too little contrast. Having done that, and I'm not going to show you images of these because they get really ridiculously silly graphics, but you have to extract from the image just the chest area where the spots are. And you've got to get that in such a way that it's fully frontal, you can see right across the chest and you know where the boundaries are. And then it gets even more technical. You have to extract the pattern. And the pattern you extract is, for those of you who understand a bit of mathematics, you extract the set of all the vectors which join every spot to every other spot. That is a lot of data. And then you have to say, well, that's not accurate because the bird can twist and move around a bit. So you have to allow an error on that, depending upon the length of those vectors. And then you have to have a set of algorithms which go and say, is this penguin already in the database? You have to go and search through that. And then you have to say, each of those steps takes time and computer power. It is a much more complex process than trying to do facial recognition in humans, because at least in humans, the distances between the eyes are fixed. The distance, the length of the nose is fixed. All those things are fixed things. Penguins' chests are not fixed. They can move around. And also they can have dirt, which obscures spots. They can have horrible bright reflections, which make spots not visible. So it's a much more computationally difficult problem. And then it can be done. But the real problem is that if the number of penguins gets too big in your database, the time to process the things gets prohibitively long, and you will not be able to analyze the data coming from a single camera in the time it takes before the more data arrives, so that you will never be able to keep up with the data input. And that gives a size limit to the system, unless, of course, you go to supercomputers and spend thousands and thousands and thousands of dollars. So basically, the last time we worked it out, which was probably now about 5 or 6 years ago, the system would not be suitable for populations bigger than about 1,000 birds. And that means it will never be able to replace banding, because banding on African penguins, I mean, as the population of African penguins falls, of course, this becomes less of a problem, but the number of African penguins that are banded currently is around, that have been banded ever, is about 90,000. Most of those, of course, are no longer with us. So that's no good. Which brings us to the only solution we have that actually works incredibly well, which is to microchip the penguins. So these are using standard pit tags, used for pets. Actually, the ones we use are designed for fish, but it doesn't really matter. They are used, they're quite small. The ones we use are about the size of a grain of rice, a little bit smaller than the average rice grain. And they are very reliable. They don't weigh much, so they're just injected into the penguin's with a flap of skin just above the penguin's foot. They can be read at nest with a handheld reader. Here is handheld reader reading a penguin. You need to get the end of that reader to within about 30 centimeters of the penguin's foot, and it will read it. They're reasonably non-intrusive, and anyway, when one is monitoring, one often uses what we call a penguin stick, otherwise known as a broom handle, to lift the penguin up to see what's underneath it. So this is no more intrusive than other monitoring. And also, of course, we can set up readers along pathways which are fixed, and just the penguin walks over it and it's read. Now, in about 2013, clipper banding of African penguins was stopped. It was deemed to be too dangerous, and we had to stop it. Since then, about 10,000 African penguins have been fitted with transponders, and more than 125,000 readings have been made of those birds. Of the birds that have been fitted with transponders, around about between half and 60%, I can't remember the exact number now, have been seen at least once. If you compare those numbers to clipper bands, it's much, much, much more efficient. You get much better data because it's electronic. You don't have to worry about people misreading it, and the data is, you don't have to go and get it. Most of the data actually comes by sitting at home and waiting for the readers on the islands to send you an email every day telling which penguins have crossed it. So that's the current solution. Use microchips and get the data that way. So that's all I have to tell you, I think, and I hope you've enjoyed listening to it. Thank you.
Fantastic, Peter. Thank you for a brilliant presentation. And yeah, I think it very early on in the presentation, I think it was quite easy to understand how your love of penguins started and how it's continued and is still continuing. So thanks very much for that. A fascinating account of the history of marking penguins and and the reasons for the changes. It seems that the transponder idea is a very, very good idea.
Can I guess that the student was Kate, who described penguins as two razor blades or not?
No, you're much too late on. It was Phil.
Okay, Phil, yes,
Wow.
excellent. Yeah. That's a brilliant description. They never told me that.
Yeah.
Written their thesis.
Yeah. There's been a little bit of chat on the chat group about Peter the penguin. I mean, that was a fascinating story with Peter, Pamela, and Percy. Watching them travel back from Port Elizabeth to their islands off the west coast.
Peter, you better do a quick recap on Peter.
Okay. Okay. I mean, what happened was, I mean, this was before I was very much involved in South Africa, because this happened in 2000, and our project on Robben Island and Clipper Vans started in 2001. So it was, I was at that time what you would call a rank amateur. But what was done, if you remember, there was about roughly 20,000 birds were oiled from Dassenau and Robben Island, taken away to be cleaned. Then more oil was going toward Dassenau Island, and there would have been another up to 20,000 or more penguins oiled more. And frankly, there was no capacity at all to be able to deal with more oiled penguins. So a decision was taken to box them up, ship them to Port Elizabeth, and put them in the water there in the hope that they would swim back safely to the Western Cape, and that the time it would take them to swim back would be sufficient for people to clean up the beaches around the islands. I don't think anyone was at all certain what would happen. There is, yes, we know that penguins can move around quite a lot, they can swim long distances, but no one's actually taken an adult penguin 600 kilometers away from where it should be and put it in the water and know that it will swim back to where it came from. So it was a good idea to put some trackers on these birds. As I understand it, the people who were doing this, the Department of Environmental Affairs, borrowed, in inverted commas, some satellite trackers, which were supposed to be going on seals, but they were in someone's office, and so one of the people working there went and borrowed them. They were put onto 3 birds, which were released at Port Elizabeth. The first one that got the name Peter for the very simple reason that when they were sent off, one of the staff from Environmental Affairs, Lisa Upfold, as she was then, was briefed to put the tracker on and then to in front of the press. Great, having to do this job in front of the press. Had the photographs taken, then answer any questions. She got only one question, which was, what's the penguin's name? And the first name that came into her mind was Peter. So that penguin was called Peter. That's how he got his name. Then he was put in the sea, and then he was sent to swim back. And every few hours, a position came from the satellite data, which went up to France and back down again. And people basically put them onto a map, which was dynamically available to anyone. I remember seeing that dynamic map on the BBC News at lunchtime when I was in the UK. So I knew it became quite famous. Peter was on all the placards along here, you know, Peter Penguin reaches, paper gallows, Peter Penguin almost, you know, home, Peter Penguin home. When Peter Penguin got back to Robben Island, which is where he went back to, his transmitter kept working. And, you know, these were valuable things. They were meant to belong to these seal researchers. So a team of people went out on Robben Island and searched for him, and they knew where he was about 3 or 4 hours ago, because that's how long it took to get the fixes. And they went onto Robben Island to find him, and I think they spent in total about 2 weeks before the transmitter packed up, and they kept looking for him, and they never actually managed to catch him. So he then was basically a missing bird. He did, of course, have a flipper band on, a ring, A14059. And he was found purely by chance because, well, when we were working on Robben Island, as we were regularly, we always read the bands as much as we could. And it was 3 years later, I think, I was walking along the shore with a volunteer and a couple of other people. With my telescope, I saw a bird out on the rocks. That's where the picture was taken, and put the telescope up and managed to read the band, and then called over everybody else who was anywhere near me to come and read the band as well to confirm I was right. And then we told people. The only problem with that was the date. It was April the 1st. Nobody believed us. We had to get that photograph. So anyway, that's the story of Peter. He hasn't been seen since either.
Well, that's a,
Thanks,
a
Peter.
wonderful
That's
story.
fantastic. Yeah.
Yeah, fantastic. Yeah, just everyone saying thank you very much for a wonderful presentation, Peter, and for sharing this with us. I think I've just got one last thing to ask you to do, and that is to stand up and show us what's on your T-shirt, because I think just a few of us who were chatting before we started. So there's the penguin, or some penguins, as Peter was saying. A lot of Peter and his wife's lives have revolved around penguins. So thank you for that. That's wonderful. Thanks so much for sharing an obvious passion for penguins, and adding the professional aspects of your work and your career into that as well. Yes, Itasa.
There's a question from Jonathan. He's asking, why do the wild birds, penguins, learn how to take the rubber bands off while captive birds?
Yes, it's a very good question. In captivity, they just leave them on. There's no problem at all. We had them on birds in Bristol Zoo for 1.5 years. Not a single one came off. In fact, all the birds in Bristol Zoo, and I can think of 5 or 6 other zoos, have got those bands on. And I've talked to many of the keepers, and they have never had a recording of a bird take a band off. When we came out to Robben Island, we started putting them on. We actually put a transponder and a flipper band on each bird. So the idea being that we could always be sure what had happened. Within 2 days, we had a bird come back with a transponder and no band. I have never seen one take one off, but I have found them in the nest on the ground. So I don't understand why the wild birds behave differently than the captive birds. It may just be that captive birds are much more used to being handled. They're much more used to having things done to them because they are regularly handled. I mean, they're going for medical checkups once a week typically. So I suspect it's just that they didn't really care so much. And also they didn't have to worry about getting food. I mean, if you're a captive bird, it's a life of luxury. You know, you get fed twice a day, you get full medical care for free, you know, you haven't got to work hard for anything. So I think that probably they behave differently. And it's an interesting example of how captive and wild behavior is quite different.
Right, thank you.
Thanks. Can I ask another question? Just got in my mind, you said you're happy with this individual pattern of the African penguins with their chest pattern. But what is with the blue fairy penguins from Australia or the other species who have not such a pattern?
Okay,
are there also ways to identify them individually, or are you lost, or are you only with a transponder?
Thank you, Jonathan. When we were doing this initially, we did in fact look at all the species of penguin. Little penguins actually have a different pattern. They have a pattern of feathers on their flippers, which have a white edge. And those patterns are actually quite unique, and they don't change. Again, they survive the molt. So there was a way of doing those as well. We found pretty much every species, with the exception of adelies, that we could find something which is variable. Tilo Burkhardt, the PhD student who was responsible for writing the system, now runs an animal biometrics group in the University of Bristol. He's quite a large group, and he does a lot of work on designing systems to recognize species. The only species that we haven't managed to crack it for, or the only group of species haven't crack it for and keep it working in real time, is penguins. Unfortunately, that's where he started, but he's got whale sharks, he's got zebras are great. Zebras are easily doable, and so on. So he's got all these systems working, all based on the same principles as the African penguin original system. Basically, the best spot system he has, by the way, which is really incredible, which is used massively in research now in the university, is on ants. is for all the people who want to do ant tracking, they used to have to paint little things on the backs of ants to be able to track them. But now you just put a camera above it, and each ant has a set of pits on its back, and those pits form a unique pattern. So you can recognise, he's got there, they have in Bristol, they've got a little ant research group which got a ant nest, I think has about 10,000 ants in it at any one time, and they're all tracked continuously by cameras and identified as individuals. So that piece of research has become quite very useful across a lot of places, but just not with penguins, unfortunately.
And do the penguins, they recognize each other via voice or also via these patterns? Or do they recognize each other by voice? Or how do the penguins recognize each other?
It's a very good question, Jonathan. Penguin adults, parents and chicks recognize across all species, we're pretty certain by voice. There's been some very good research done on that. The French actually done a great deal of work in voice recognition where they've done made recordings and played them back and so on. And it is clearly the case that all penguin species use vocal recognition for their offspring. But of course, when the offspring become adults, their voices change completely. So they probably aren't recognizing beyond that. And since chicks can identify adults, presumably adults can differentiate between themselves.
Great. I think just to say thank you so much to Helen and Peter for two wonderful presentations.